Bismillahirrahmanirrahim. Assalamu alaikum. Experiment number 7 is about the adsorption using the granular activated carbon. The activated carbon is used to treat the wastewater and it can remove many type of the pollution. So to this experiment having the objective to understand the adsorption phenomena of removing color from a water sample by using granular activated carbon. As I told in the theory class, granular activated carbon is like the sand particle made of the coal, faham. So that is a special type of the material which can use to treat the different type of the wastewater. It can remove the color, it can remove the taste and odor, it can remove the many other pollutants. So the procedure is already in your lab manual. First step, for example, to prepare the granular carbon in the lab by drying this material, and then prepare a wastewater solution also, in which you can add some color, like we use the methylene blue to make a colored solution. So this, for example, this is the colored solution and this is the activated carbon we can use in the lab. And then the steps for the experiments I will explain later on. Once you will complete the experiment, then this table you have to prepare. You will collect the sample at different time intervals and you will measure this value from the colorimeter. The color you can measure, and then you will convert into concentration of the methylene blue, how much color is removed, and then you can do this calculation. So that is the one of the objective. So in the experiment, as I discussed, that we will use activated carbon. This is the activated carbon. We will use the sample of the wastewater having some color. We will use the colorimeter to measure the color. These are the keywords for collecting the sample from the solution. So this is the material and everything which we will use in our experiment. Uh, Regarding uh, the the first step, we have to take some activated carbon. We have to measure its weight. Generally we take 5 gram of the activated carbon, we using the weighing balance, and then this is the solution having the color. So what we do, we take initially some amount of the sample, like in 5 milliliter to 10 milliliter, to find out the color, how much color is in the sample, right? And then we add this activated carbon and we start mixing. We add this activated carbon in 1 liter of the solution. We start mixing. So once the mixing will be going on, you will see the color will reduce with the time. So activated carbon is there and it is mixing with the solution from which we need to remove the color. And after some certain time, for example after 5 minutes, 10 minutes, you will collect the sample and you will see with the time the color is decreasing. Ultimately you will see something like that, the color, all the color will remove. So this is the treatment with the time you will see in this step. After that, once you will get the samples, different sample at different time intervals, as I told you in the data, you will collect the sample at different time. 0 time and initially before adding the carbon, then after 5 minutes, 10 minutes, 15, and all this, you will get the sample. Then we use a colorimeter. You remember we use the colorimeter in one of the experiment. So you have to switch on. Then in the main menu for all program, you will go to the experiment number, which is testing number 104. Here you have the absorbance. You will measure the absorbance in the same way. First scan the blank by using the distilled water, then scan the sample. You will get the value of the absorbance. The value you will get from here is not the color. It is giving, the meter will give you the absorbance. So how you will measure the color? By using this graph. This is a calibration curve for measurement of the color. So what you will do, you have the value of the absorbance. For example, you get the absorbance value 0.5. So at 0.5 you will go to this point, and from here you can go to this point and you can calculate how much is the concentration of the methylene blue in milligram per liter. For example, at 0.5 you get the value of 32 milligram per liter. So this is your value of the concentration. You will use this value to fill this table. So for example, you get the absorbance of 0.5, then from there you calculated from the graph 32 milligram per liter. So for all the sample, for every time interval, you will calculate the concentration. From this concentration, then you will do the calculation, the C over C naught. C over C naught is the ratio of the concentration. C naught is the concentration at 0 time. At 0 time concentration is 32, for example. Here at 5 minutes it is maybe reduced to 30, or then 28, and then like that it will reduce. So C over C naught is the this value divided by the initial value. So for first case it will be 32 divided by 32, because it is the first value already. So 32 divided by 32, it will become 1. Then later on, whatever the value you have here, you will again divide by 32. So every value you will divide by 32, so you will get C over C naught value here, right? Then you have to calculate delta C. Delta C from this formula, Cn minus 1 minus Cn. For example, this delta C will be this value minus this value, right? Or you can say that the value which is higher, this minus this value, so it will give this value. Then again this value minus this value will give this value. So the first value will be blank because there is no initial value from here. So this, you will get this second value from here. How? This 32 minus, for example, 30, so you will get here. For again next value, for this value minus this value, you will get this value. In this way you will get delta C. So you will get this column and this column. How you will calculate this mass of methylene blue transfer per gram of the carbon? By using this formula, right? So it is the formula is the delta C, which you have calculated here, multiply the volume how much you take in this experiment, 1 liter, so it will be 1. Mass of the activated carbon, we take 5 milligram, so it will be 5. So by simplifying this, you will get all this value. Now in the last step, what you have to do, you have to prepare a graph using the two value. In the same graph you will plot the with the time, this is time on the x-axis, and C over C naught on the y-axis. So you will actually first graph will be between this value, right, and this value. This is the first graph you will make, and then second graph you will make between this value and this value. Same graph paper, right? So you will get in the graph paper, like this is the graph paper, one line may be something like that, another may be something like that, and two graph you will get, right? So here it is the time, here time, and here is the two value. One is, sorry, not this one, the C over C naught, right? So in this case, right, so you will use this value, the first graph between these two values, second graph between this value and this value. So the time will be on the x-axis, right, and the y-axis this value and this value. So you will get two graphs. So this is our experiment, and it will give you the idea with the time how the color is reducing, right, and you will see the reduction in the working of the activated carbon. And you will like in the manual you will write the results and discussion, how the color is removing with the time, and what is the end result you get, how much color reduce. So like this one you can put in the result and discussion. So that is your experiment. So this is all about the experiment number 7, which is about the activated carbon adsorption, right? So if you have any question, you can discuss with me. And just what you have to do now, you have to complete this manual. I will send you the reading of absorbance. Based on that reading, you will do this calculation, you will prepare the graph, and you will write down the results and the discussion. So this is our experiment, right? Okay, thank you very much. I will put on the blackboard the readings, the data for this experiment. Thank you.